DGIST announced on March 16 that a research team, led by Professor Byung-Chang Suh, has investigated for first time the real-time effect of G protein cycle, which acts as a switch in our body on structural changes in G protein-coupled receptors. GPCRs are activated by external signals such as odor, light, temperature, neurotransmitters, and hormones, and are involved in numerous biological activities to the extent that about half of known drugs target GPCRs. GPCRs regulate various intracellular signaling pathways using G proteins. However, role of the reversible activation-deactivation loop of G protein on structural changes in GPCRs has yet to be defined. Professor Byung-Chang Suh's research team has developed new biosensor based on a fluorescent protein that uses the human M3 muscarinic acetylcholine receptor type of GPCR. Using this biosensor, they found that the GPCR-based single-receptor sensor exhibits sequential structural transformation via the G protein cycle. The research team also showed that G protein activation causes two-step change in the HM3R structure, which includes a rapid step GQ protein binding followed by slow step of physical dissociation, the GQ and G subunits. They also found that cleaved active GQ formed a stable complex with ligand 1, activated HM3R and PLC, the downstream signaling pathway of GQ. In addition, applied research by Professor Suh's research team on pathology of, for example, pathology G protein-associated gene mutations that cause uveal melanoma 2, and pharmacology of relevant therapeutic drug candidates was demonstrated by independent binding G subunits from GQ to HM3R. Provides tips for possible treatment of related diseases. Corresponding author Professor Suh said about study, "We have verified real-time communication between active GPCRs and G proteins, which have been considered separate up to this point, and they expect to be very helpful to future molecular individual-level research into GPCRs and G protein-related diseases, their treatments." This study, the world-renowned journal Nature Communications on March 8, 2023, was attended by DGIST Brain Sciences Department mixed graduate and doctoral student Yong-Seok Kim as the first author. Foundations, Mid-Career Researcher Program, and Basic Research Laboratory.